This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 25, recorded January 12th, 2012. Hello, everyone. I'm Vincent Racaniello, and this is TWiM, the podcast that explores unseen life on Earth. Joining me today in this first TWiM of 2012 from the Medical University of South Carolina, Michael Schmidt.
Hello, Vincent. Welcome to a new year.
Happy new microbial year here on TWiM.
You know, it's funny that you say that. I think this is actually going to be the decade of the microbe. I think we are going to set, or the microbiologists working in this field in concert with our colleagues in systematics are really going to set everything that we thought about microbes and hosts on its ear.
Gets better and better, doesn't it?
Oh, yeah.
Also joining us today from Small Things Considered in San Diego, sunny San Diego, Elio Schaechter. Happy New Year.
Howdy. It's not exactly sunny today.
No?
But almost. But on the other hand, I go out in shirt sleeves at this time of the year, so I can't complain.
Well, here, Elio, just to let you know, it's miserable. It's cloudy, it's cold, it's blowing wind, and it's pouring rain.
Well, come on down.
Yeah, really. I shouldn't complain because I'm here because I like it here. But it is a contrast to where you are. Nonetheless, did you have a good New Year, Elio?
Yeah, we went to the desert. We have a place in the desert, and we had some family there, and it was nice and pleasant. And so, yes.
Is it really desert? There's cacti and sand and...
Cacti and sand, absolutely. It's real desert. It's the Great West, the end of the western end of the Great Western Desert.
Nice. It sounds pretty good. Is there fireworks on New Year's Eve?
I wouldn't know because I declared that midnight was at 9 o'clock for me. So.
Yes.
We saw the ball drop and went to bed.
There you go.
That's the advantage of living on the West Coast.
Right, exactly.
Ah, yes, right. You get a 3-hour jump on the ball. Huh? It must be strange.
But I'm complaining to you virologists.
Yes. What did we do?
I have a cold, a common cold. I've had it for almost 2 weeks, and I wish you guys would get off your butt and stop studying all these esoteric viruses and cure the common cold.
Elio, you know, we actually do work on rhinovirus in my laboratory.
Uh-huh.
We have been doing various things, and in fact, I have a student who is trying to take some antiviral approaches. So who knows?
Maybe.
We tend to be academic, unfortunately.
Actually, let me ask you a question.
Sure.
The two of you. The most common human condition that leads to misery is vaguely called malaise. Malaise is what you have when you have a hangover. Malaise is what you have when you have a cold and so forth. And it's miserable. And it's all due to cytokines. It's sort of cytokine poisoning, isn't it?
That's right. Yeah,
and I don't know why, I don't understand why billions of dollars are not spent in trying to get rid of the cytokine, the extra level of cytokines that makes you so miserable. How about inventing a drug for that and we'll all be rich?
Well, those cytokines are needed. Right?
I know, but not in smaller amounts.
I think the problem is you're trying to close the barn door after the horse has left. Uh-huh. And, good old non-steroidal anti-inflammatories can actually mute the cytokine response. So if you have been exposed to someone who has a active communicable virus, you may just pop a couple of garden variety non-steroidal like ibuprofen, and you may be able to suppress the cytokine response and not feel as miserably. You still have a viremia, and your immune system will still have to repair the damage, but you'll actually mute the cytokine response that makes you feel so miserable. But I think it's Mother Nature's way of getting you to rest and drink plenty of fluids. Because
yes, doctor. Yes, doctor.
Well, no, I mean, it actually works. I mean, I'm not a doctor. I only play one on TV. And it really is remarkable how those old wives' tales actually do indeed work. They do make you feel better.
It's a good thing we record on separate channels, so every time you cough, Elio, we can delete it.
You can bleep it out. Very good.
All right, today we have two amazing papers, or two amazing stories, I should say, and they are completely different, but we're going to put them together for TWiM 25. And the first one is something that I'd never heard of, but is incredible. You're going to love this. It's all about magnetotactic bacteria. Elio, this is amazing.
Okay. This is part of it goes like under the heading of gee whiz microbiology. There's so many aspects in microbiology you can point to of things which are totally unexpected, totally almost unheard of, and this is one of them. So here's the story. Let me start historically. In 1975, a microbiologist by the name of Richard Blakemore was working at Woods Hole, and he had some pond water, I think it was, or seawater, I don't remember, some water under the microscope, and was noticing that a lot of bacteria were swimming in one direction. Now, that's unusual. When you look at your ordinary bacteria, they swim in every direction. And so he was wondering what caused that. And he had the smart idea, and really the great insight of figuring out that maybe this had something to do with the magnetic pole, with the magnetic current of the Earth. That is, there's a magnetic field on Earth which goes from the pole to the pole, and that is significant. It's not very strong, but it's significant. And maybe the bacteria were listening to that force. So what did he do? He took a magnet and put it on the slide, and the bacteria, all of a sudden, depending on which way he oriented the magnet, either swam towards the magnet or away from the magnet. And he looked at this bacteria. By the way, this bacteria, dead easy to cultivate. All you have to do is to take some mud, some slurry of a mud, put it in a test tube, put a magnet on the side of the test tube, throw out all the liquid except in what sticks are going to be magnetotactic bacteria. No, not hard. Anyhow, it turns out, and this is worth saying, that in fact, this had been discovered by an Italian, Salvatore Bellini, a professor at the University of Pavia, who had found exactly the same thing. Only the poor guy had published this in some ultra-obscure Italian journal. Nobody knew about it, but luckily this story has been unearthed by one of Blakemore's colleagues. And of course, Blakemore gets the credit for having done the experiments in a visible way and having continued and making many contributions to this field.
Isn't it amazing that he had the insight to see them swimming in one direction and then do that experiment?
I think it's amazing.
You have to be so observant.
Exactly. This is really good science. I mean, this beats a lot of complicated stuff that we deal with. I really admire it.
And to give our listeners, those of you who may be high school biology teachers, these bacteria are distributed across the planet. And so you can go out to your favorite pond and get some mud with some water, and you leave it settle for a while. And then you can get your garden variety magnetic stirring bar and get some modeling clay and put the stirring bar just above the mud-water interface. And you can literally watch the bacteria swim out of the water, and they'll accumulate a turbid pellet at the side of that stirring bar. The first time I saw this as a graduate student in 1980, I was shocked and amazed. And you then just take your little skinny Pasteur pipette and you can aspirate them off, and you can put them under a microscope slide, and you can actually steer them just by moving your garden variety stirring bar around. So if you got access to a good old garden variety pond, and I know many high school biology folks do actually go out and do pond water, you can just actually pick them up and you can isolate magnetotactic bacteria from almost any pond that you stick a bucket into.
Isn't that neat? Yeah. So now let's figure out what this is supposed to be all about. So the simple idea is the following. These guys are microaerophilic. They don't like too much oxygen. In fact, they hate it. They're not totally anaerobic. They need a very tiny amount of oxygen. And in columns of water, in lakes and in the ocean and everywhere, is a zone called a transition zone of oxygen and anoxic conditions. And that's what they want. So they are trying to go there. And this usually is deep. It's a ways down from the surface. So if the bacteria find themselves up in the surface or nearby, their job is to find this zone, this transition zone. Now, the way the magnetic field works is that it goes across the Earth, let's say, from the pole to the equator. So by the time you are at the equator or at medium latitudes, the field makes you want to go downwards if you are trying to go along, to align along the field. And this is what happens, so that in fact, well, this is the theory, that this is the reason why they are magnetotactic, because they use the magnetic field of the Earth to line themselves up along, to go down the water column.
I refer to this as the Goldilocks gene, because the Goldilocks gene allows these bacteria to find that right zone where the oxygen tension is just right. And it's the Goldilocks gene.
I just got it.
Terrific.
You thought I was crazy, but that's exactly what it is. And they come up with a complicated name. They call it the oxic-anoxic transition zone, or OATZ. I think most people, OATZ. So it's OATZ, or I just referred to it when the first time I thought about this, I said, well, this is like, is this bed too soft? Is this one too hard? This one is just right. So it's the Goldilocks. And this is effectively what our friends in astronomy are doing. They're looking for the right planet. Well, we got bacteria that find the right level of oxygen just by using the magnetic force that comes out of the planet.
Now, that's true, and that's the standard view of the thing. However, this being biology, there are always exceptions. There are always things that interfere with a clean, simple statement. And here's why I say that. Well, first of all, it looks very good, because if you isolate this bacteria from the northern hemisphere, they tend to go towards the north. And if you isolate from the southern hemisphere, they align with the southern magnetic field. That's very good. However, you find magnetotactic bacteria at the equator, where there is no selective advantage. So what are they doing there? We don't know. Also, this is nasty, you find south-seeking magnetotactic bacteria in the northern hemisphere. Now, what are they doing? So this does not deny the principle that we just explained, that this is a way of seeking that magic zone of the right oxygen concentration by using the magnetic field. But there's more to it, or let's say some bacteria do this, other bacteria use their magnetic properties for other reasons. So we'll come back to that later. Let's get a little bit into the subject matter first. So if you look at this bacteria under the electron microscope, you find that they have little crystals. These crystals are made up of iron in the form of magnetite, which is iron oxide, or greigite, which is iron sulfate. And these crystals are in a regular, arranged in a regular form in a chain. So you find a chain of 10, 12, up to 100 of these things, depending on the strain. Okay? And they are about 70 nanometers across, and they are encased in a membrane, which is what we're going to talk about for a while. The encasing in the membrane makes it a little bit interesting, because this becomes a model for organelle biogenesis, if you wish, if you want to consider this is maybe the equivalent of organelle. So anyhow, they have a magnet in it, which is made up of a chain of little tiny magnets. That's all it takes, because if you use dead bacteria, they will align themselves also. So the magnet is really the thing that does it. Let me first start out by saying something that Mike can probably pipe in and help me. The biotechnological uses for this. One of them is that these bugs make very even crystals, and for people who care about that, that's a big deal. They also can be introduced into animals, people, and can be used as markers in an MRI analysis, magnetic resonance, because it detects metals.
Huh.
They can be used for other things too, like separation of cells. If you introduce this bacteria with these magnets into cells, you can then pull out the cells with magnets. You can shoot these guys into other cells, and you can certainly put some DNA on the or so anything you want. And this is one way in which you could make cells magnetic. Well, the problem with biotechnology right now is that most of these guys cannot be cultivated, and if they grow, they don't grow very well. And so you want to clone them into E. coli, but that's not been all that successful to date. So we're waiting technological developments of the sort. I don't know, Mike, have you run into this in terms of applications?
The lab I grew up in when I was a young graduate student was working on magnetotactic bacteria. It was
You're kidding?
No,
Where was that?
I was in Walt Konecka's lab at Indiana University.
Oh my goodness, yes, big iron guy.
And he when Blakemore's paper was published in Science in 1975, he got one of his new graduate students to work on it, and this student's name was Tom Mensch. And Tom
was he?
Yeah, Tom actually was a really good guy. He was a Mensch. He had finished his PhD just as I came into Walt's lab, and he spent his career, and then this was the PI's projects, Walt's project, to try to figure out how to get these microbes to grow in pure culture. And we had a lot of discussions about the applications of using these tiny little magnets. And, this was before the great revolution of the electronics era. And, we thought of getting uniform particles for magnetic tape and magnetic discs because it was so uniform. But, we didn't imagine the MRI at that point in time because this was 1980 and the MRI was still called nuclear magnetic resonance. And so I think there are other tremendous applications for this, and the paper actually alludes to this in terms of this magnetite in a string actually serving as a small little battery in which the magnetic force lines of the planet are actually generating a current. And if you think about it, if you could put magnetotactic organelles into your cell phone, such that as you carried your cell phone around, it would generate a trickle charge to continuously charge your battery, you truly have the golden goose of being able to move this into the truly mainstream. And, this review article that we're discussing this remarkable biology from, they really do a phenomenal job laying out some of the most intriguing aspects of this microbe. One particular quote from the paper is, the organism can then exploit the change in the redox state of the mineral to generate energy. And I remember having a discussion with Norm Pace. Norm was at Indiana at the same time I was, and this was in the halcyon days before 16S when we were doing 5S sequencing to identify microbes, and we were looking at freshwater magnetotactic bacteria, and we had to isolate enough of these little bacteria from the mud to give to Dr. Pace so he could figure out our 5S RNA sequence. So I have fond but frustrating memories of trying to figure out how to grow these things. It was sort of the lab's project to try to figure this out.
Well, if it's any comfort, it's not that easy. And so even though it holds a lot of promise, it still hasn't panned out. So you didn't miss out on anything yet. There's still room for that.
Elio, what's the taxonomy? Is there just one genus that is magnetotactic? Or there?
No. This is fairly widespread. It's all gram-negatives, and some are cocci, some are rods, some are curved, some are helical. Most are proteobacteria, alpha and delta. But there are, it's fairly widespread. There is even a really most interesting bug that a Brazilian by the name of Lins has studied, which is multicellular. It looks like a like a collection of round cells put together, something like a blastula, if you wish. And this is highly structured. This is not your usual staph arrangement or multicellular things. This is a very structured thing. And if you try to tease the bugs apart, they will not grow. So it looks like the only case I know of an obligatory multicellular bacterium. Bacteria can be multicellular plenty, but that's usually something that may happen or not. Here, it seems to be obligatory. And they are magnetotactic, and it's really sort of something all by itself. I should give you a paper to list, because this is really makes good reading. And there's a picture in the review article we're talking about that shows you that, and it's just amazing. But as I say, there are quite a few different kinds of bacteria. Now, and most of them have not been cultivated.
Can I ask you one more thing? So this model that the magnetotropism is important for them to get to the right zone where there's just the right amount of oxygen. Does that imply that most of them are in aquatic environments?
Or, yeah.
Yeah?
Yeah, they're isolated not routinely from aquatic environments. Whether they exist elsewhere, I wouldn't doubt it, because there is water in soils at times. But I don't know that this is. Do you know, Mike, if they're found?
No, I mean we speculated a lot when I was a graduate student is if you could isolate them from dirt. And so it was the running joke in the lab of where do you get dirt that would have magnetotactic bacteria in them, because our selection for these microbes is they had to swim out of the sediment. And so this was before the days when, magnetic, now magnets are pretty common in the modern laboratory because immunologists have exploited magnetic principles to do cell sorting and other things. And so we could never do those experiments simply because we didn't have the technology and we were focused on other things. And it was one of those side projects. But I think if I was a betting man, I would actually bet on finding them in dirt, especially if some of these other hypotheses that the author of this review, who works in this field, actually speculated on, especially if the magnetosome battery, where, the fundamental issue in microbial physiology for all microbes, especially those that live without oxygen, is where to dump their waste electrons. And if they are able to achieve a redox benefit from the magnetic field to effectively make reducing equivalents without burning carbon, to take the carbon all through its oxidation state, that will actually confer a fitness advantage because it's all about reducing equivalents. And if you don't have to burn carbon and generate those waste electrons that you then have to dump, where you don't have the luxury of molecular oxygen, I think that could be a tremendous selective advantage to keeping something like magnetotaxis.
So this says that, more than that, it says that being magnetic does not necessarily refer to magnetotaxis, but to other phenomena, like Mike just said. So there's room for, there's a big tent here. There's room for a lot of physiology. And by and large, most of it is still, I think, unexplored. We tend to do the easy things, which is these days to look at genes, look at genomics, look at proteomics and stuff like that. But the hard-nosed physiology and the ecological physiology which is relevant to the environment, still needs to be done. Let me point out, however, this is a point that I'd like to make, is that these guys are important in the following sense. In the ocean, they are, or in many waters, let's say, they are about 5% of the total bacteria, so they're abundant. They they contain within them between 1 and 10% of all the iron in sediments, so it's a lot. In addition, they've been found as fossils because the magnetite is retained in a fossil and can easily be seen. So this is really a spectacular type of paleomicrobiology. They may be as old as 1.9 million years, which is pretty old, and they may have arisen when oxygen started to come into the atmosphere. But they're probably the first biological use of magnets. Today, of course, we know that magnets are found in birds, in insects, in algae, and even in human beings. And by and large, we really don't know what it is they do. They just, who knows?
In humans too, we have magnets?
Yeah, we have some magnetite in us, and nobody knows what it's about.
So what happens when you go into an MRI?
It's too little to count.
Okay.
It doesn't
So
matter.
here's a hypothesis. I came up reading this, and I was thinking, because I have to give to my dental students the antibiotic lecture in a few days. And so here's my question. Are bacteria that effectively sequester large amounts of iron, because that's what these magnetic bacteria do, the equivalent of antibiotic producers in the environment? Because antibiotic generation is effectively a tremendous selective pressure to carve out that niche for a microorganism. And in a sense, biologically available iron is always limiting, because now we live in an atmosphere with oxygen. It turns to rust. And since the Fe+2 is always limiting, by virtue of the fact that the magnetotactic or the magnetic bacteria can concentrate iron, is this a precursor, or are they just, like our friends, the streptomyces, that make antibiotics to control their niche so that it gives them a fitness advantage?
Well, this makes sense because of the transport mechanism. To make a magnetosome, that is to make the crystals, the iron has to be transported in. And in some of the bugs that have been studied, the rate of transport of iron is 100 times faster than in E. coli. And they use siderophores, which are small molecular weight compounds, which are chelating agents, essentially. These are the hydroxamate type, for those who know. They send out these essentially messengers to sop up the iron at high avidity. And then they have mechanisms for bringing in the siderophore with its iron cargo. And as I say, these guys are very, very good at that. So you may be right that this is a way of keeping the iron from other bugs. It may be a way of coping in the environment. Sure.
Michael, are these bacteria preyed upon for their iron?
I don't know. That's a really good question, Vincent.
It's a great question.
Well, what argues against it is the fact that they're found in fossils. Because if you could steal the iron once it becomes magnetite, then you would almost think that it would be involved in, there would be a circle to that mineral cycle. But by virtue of the fact that they're shown in the fossil record, and that just may be a function of how quickly mud was made into rock, that would argue against a true cycle where somebody else can steal the magnetite away.
Could be. But it's still a possibility. I think, for one thing, I have not seen anything said about viruses. There must be phages that affect these bugs, and I haven't seen anything about them.
Well, given the fact that they're aquatic, you would almost bet the ranch on that.
But I don't think that has. But there may be a literature on it. I haven't seen it, but that doesn't mean anything. So if there are viruses, we don't know what they do. Anyhow,
what suggests that there may be viruses is by virtue of the fact that the genetics that controls the formation of the magnetite is on, for lack of a better term, the equivalent of a pathogenicity island, which is a hallmark of being moved by phages, and they actually coined the term of a magnetosome island. It's got all the same hallmark features that pathogenicity islands have. Different G+C content. It's associated with tRNA genes, pseudogenes, transposons, and IS elements.
Yeah. In fact, this island is ubiquitous among the magnetotactic bacteria that have been studied. The genes are highly conserved. It's about 98 kb, and it codes for about 100 proteins. So this is not a small thing, and it does suggest that lateral gene transfer may be the way in which this thing goes from one bug to another. And it's possible that when you acquire this island, you acquire the whole schmear. All of a sudden, you are able to make magnetosomes and become magnetotactic. So this is really quite remarkable.
I wouldn't be surprised if some phages didn't package some magnetosomes to allow them to home in on the bacteria.
That's cute. My goodness.
I think the virologists have to get busy, Vincent.
Yeah, just easy to purify them with a magnet, right?
That's right.
Yeah.
My goodness. That's how about it, folks out there?
Yeah.
Somebody's stepping up to this one. It's really great fun. All right, so let's talk a little bit more about the bugs, the ones that have been studied. There's sort of a model system, Magnetospirillum. Turns out to have a very high number of chemotactic proteins. It has 60 methyl-sensing chemotactic proteins, methyl-dependent chemotactic protein. E. coli has 5. So if we go back to what we did a couple of weeks ago when we talked about the social IQ of a kind of a bacillus called Penibacillus, we talked about the work of Ben Jacob in Israel and his colleague in Groningen in Holland, Ingham, and we talked about how Ben Jacob has a scale of a social IQ. In other words, the social IQ is determined by the number of proteins involved in signaling, communication, and so forth. So by that criterion, these bugs have a high social IQ. So much for that. Let's talk a little bit about how the magnetosomes are formed, because this is really quite fascinating. So a magnetosome is a membrane-encased, rod-shaped structure containing these crystals. It's formed by the invagination from the cell membrane, the cytoplasmic membrane, and it stays in contact with that so that the inside of this structure is in contiguous with the periplasm of the bacterium. So they're not contiguous with the cytoplasm, but with the periplasm. I hope you can picture that. Just imagine a membrane butting into a cytoplasm, which, by the way, is fairly unique. I mean, Mike mentioned that this may happen in photosynthetic bacteria. This was before we got onto the air. But there are not many cases where you have this invagination of a cytoplasmic membrane in bacteria. So anyhow, that happens by virtue of certain proteins which tell the membrane to do that. And once you have these structures, the transport of minerals and their crystallization takes place. And as I say, this takes place at a very high speed compared to E. coli, very high, strong transport mechanisms. And this is all redox dependent. And these guys depend on certain proteins, as I say, to make this structure. Once this structure is made and there's enough of the mineral there, enough of the iron there, it's assembled in crystals in a chain. And this is done along a dedicated cytoskeletal structure. It's called the magnetosome filament, which is the protein is called MAMK, M-A-M-K. This is a filament which tells the magnetosome where to sit in the cell. Even though it's still connected with the outer membrane, it could be floating around someplace and move around in a random way. It does not. This is very linear, very much along a line, and the line is due to the connection between the magnetosome bag, if you wish, and a protein called MAMK. And MAMK is a cytoskeleton protein. It is actin-like, just like some other bacterial proteins like MREB. It makes filaments in an ATP-dependent manner. And when you have a mutant in it, the magnetosomes are not along the center of the cell. They're scattered all over, and the bags are all over the place. So this is clearly a protein that's necessary for the proper placement. And the proper placement here matters, because if you had little magnetosomes scattered all over the cell, you're not going to have a magnetic moment. You're not going to be able to line up against anything, because each one is going to go its own way. So you have to have them linear, just like a magnet, just like in a straight line, so to speak. And so the magnetosome filament is necessary for that, which is fascinating, because this is one of the best uses I know for a cytoskeletal protein in bacteria. The whole subject of cytoskeletal proteins is very, very common now, a very active field of study. But in truth, there is still lots of questions about what they're for and what they do. But here is a case where we can say MAMK is there to make a filament along which the magnetosome lines up. So this is pretty good stuff. There's other proteins, lots of other proteins are known. One called MAMJ connects the vesicles to the filament. So you have to have a way for this filament to act, and this is a way in which it happens by another protein. And there's oodles of other proteins. I don't think we want to go into it. They're being studied, and there are lots of mutations and so forth. So all that is, I think, pretty good stuff.
There's a beautiful picture in this paper of the magnetosomes with and without MAMK.
Oh, yeah.
And you can see they're all lined up with and without MAMK. They're just all over the place. It's just beautiful.
It is.
So how many genes do you need to make magnetosomes? Do we know?
Well, I don't think anybody knows, but there are about 100 in a magnetosome island. So I suspect that most of them are needed. It would take quite a few things to do it. I mean, imagine each one of these steps requires a certain amount of protein work. So it's not surprising that this would be a multi-protein activity, and it's therefore complicated. But complicated or not, this becomes really a beautiful model system for studying organogenesis or something like that.
And I think that these in this review, it's really a primer of how to investigate the unknown, because what's highlighted is they used a proteomic approach rather than a genomic approach to get at the genes that were responsible. They isolated the proteins that were associated with the magnetosome, and then they effectively figured out what they were from a protein basis. And then they went fishing, because now, of course, we can sequence the genome lickety-split between the time you walk in and the time you have lunch. But what they were able to do is they were able to figure out the protein and then go on a hunting expedition, translating the frames. And they were able to discover this island. And some of the things that I think our friends in the pharmaceutical industry may be going after is, you think about the siderophore that this creature has to make that really sucks up iron. And remember, this is a diffusible molecule that goes out. And if you're looking for new ways to control growth of pathogens, sopping up all the iron is a good way of starving an infection out. And so if you could target the siderophore delivery to focused infection, and you think chronic prostatitis or prostatitis or any of the other localized infections that we have, it may be a really neat strategy that's coming from something that most clinicians would say, so the bacterium has a magnet. Who cares?
Right. Good point. Great point. All right. So left to discuss is how these magnetosomes apportion themselves in cell division. This may not be an obvious question, but when you think about it, here's a rod inside of a bacterium. And when the cells divide, you would expect each daughter cell to have a half a rod, which is exactly what happens. So you need to think about it a moment, because this is a stiff rod. It's a bunch of crystals of iron strung together. And how do you break it? And where do you break it? So this is the second paper that we are going to discuss today. It's from the lab of Scheller in Munich. And what they found is the following. First of all, when the cells divide, you find, in fact, right before division, that the magnetosome is lined up across the site of division. In other words, it's the middle of the rod-shaped cell. The same goes for cocci, but in rod shapes, it's easier to talk about rods. So you picture a rod-shaped cell and picture the magnetosome is sitting in the middle of the cell, because that's across the division site. So now, how do you break a magnetosome? It turns out that this bacteria carry out a strange form of division. Not all that strange. Mike will remember we'll know soon what it is I'm alluding to. But these cells snap. Instead of dividing evenly across, if you think of an E. coli, when it divides, the two cells just come apart. Here, the cells grow by making an angle to each other. The two daughter cells make a V-shaped structure, and they snap apart. And the snapping force is enough to break up the magnetosome. Okay, so now the magnetosome has gotten broken into two halves. One goes to the right, one goes to the left. This phenomenon, the snapping thing, requires an asymmetric kind of growth of the peptidoglycan that goes into making the cell septum. Because if it were uniform, it'd just be just like E. coli. But if you have if you wish, a factory for making peptidoglycan located on one side of the cell and not the other side, that side will grow and will make more membrane, and pretty soon it will give that wedge shape, which is what leads to the snapping apart. Now, that's neat, and it does say that there is movement of the magnetosome, because at that point, when you look at the two cells which is just divided, the magnetosomes are going to be at the poles of the new division. When the cell divides, it makes two new poles, and the magnetosomes are at that end. But the next division is going to be in the middle of the cell. So now the whole magnetosome has to travel from the pole of the cell to the center of the cell, which is a new site of division. And this says that there is some mechanism for doing that. And our friend MAMK, the protein that I said what makes this filament, must be involved in some fashion. How this happens, just how this cytoskeletal protein tells the magnetosome, come hither and locate yourself in the middle of the cell from the pole, is still not known, but it's a fascinating mechanism.
And I think they speculate as to how much energy is actually required,
right.
And it's like 10 piconewtons.
40 piconewtons.
Oh, excuse me, 40 piconewtons.
Yeah, not much.
But from the perspective of what's going on inside a bacterium at that level, to contain that amount of snapping, and any of us who have ever played with stackable magnets, separating them apart is not a trivial task. And so the amount of energy on the scale of the bacterium, you almost have to ask the fitness question, because if it impacts on the fitness of the bacterium, there has to be some selective advantage to maintain a magnetosome.
Sure.
And because 40 piconewtons, I think is, I started, Elio gave us this paper at lunch, and so I haven't had time to do my dimensional analysis to convert piconewtons into ATP molecules. So I don't... Which,
what they say, let me say what they say, though. They say that the force that's imposed on the cell by this asymmetric making of the septum is so much that it does account for what's needed to separate the magnetosome. That's what they say. As I say, I don't know. But if you look at there's a figure in their paper which shows you their schematic diagram, which shows you that if you invaginate from one side of the cell, you make for a lot of asymmetry, and that asymmetry is a lot of force. And so they say that that's enough. I mean, I have to take their word for them.
And it probably is not going to impact energetically, because if we go back to the review article, there's arguments that made as to this may be an evolutionary neutral phenotype in microorganisms, that it doesn't confer any negative selective pressure by having the magnetosome in it. Unlike pathogenicity islands, which are often frequently lost if there's no selective advantage conferred to the bacterium, because carrying extra 100 kb of DNA does indeed cost energy, and it does impact on fitness.
Could be. That's possible, but the author of the review points that as a sort of an extra possibility added to very many others for the role of magnetosomes. I'm not sure I would go there.
Yeah, it's a hypothesis that we throw out to our listeners. If you're curious, you can take it off. Take
but
up
let
the
me ask you something. Let me castigate the authors for making a statement that I didn't understand. They say, the Schiller paper, that this may be a new mechanism of cell division, the snapping apart. Well, Mike, I don't have to ask you what bacteria do you know that divide by snapping apart instead of just separating? How about the diphtheroids?
Yeah. Well, I mean, it's the first way you learn how to categorize diphtheria. It's Chinese-like characters, the
That's right.
kanji, which is, at oblique angles.
That's
And
right.
that was, when I took my first microbiology class and looked at Corynebacterium under the scope, it really did look like snapping cell division.
Exactly. They snap. And they,
Yeah.
the authors here have a movie which looks, as far as I can tell, the same way as diphtheria. So anyhow, they missed something because they didn't take your course in medical or dental microbiology, Michael. They would have learned this. Anyhow, but that's a minor point. I think this is a fabulous subject. I think we are lucky to be living at a time when work of this sort is being done and where mechanisms which are very, very complex are becoming amenable to study.
I think this paper is amazing. The images are great, and the movies are quite nice too. There's some stunning pictures of this snapping and the alignment of the magnetosomes. Just beautiful.
Right.
I mean, as we were talking about earlier, the things you can do with imaging now. Nice.
The age of imaging, I called it.
Well, I mean, that's after all what really set microbiology apart at the beginning of the last century is, the evolution of optics. We developed optics, and I think anyone who's ever seen Nomarsky optics, looking at bacteria under a microscope, their jaw drops. And now some of the imaging techniques are really jaw-dropping as well. And we should commend our friends in the optics world for helping us develop and seeing the unseen.
Let me give you a quote. You may know this. Richard Feynman, the great physicist.
Oh, yes, the famous physicist.
When he was asked about how should one study biology, he says, look at the damn thing. And by then he went on to say we need better microscopes. And guess what? This is exactly what's happened. I mean, the progress in optical microscopy, which has totally demolished the myth that there is a limit to the resolving power due to the laws of physics, all that went by the wayside by using very clever gimmicks. So that now we have resolving powers which are 10 times better than they were using this an optical microscope. Leave alone the progress in cryotomography with the electron microscope, which lets you look at really essentially untreated cells. So I think this is indeed the age of imaging. I wrote something about this in Small Things Considered some time ago.
The age of imaging. Well, on that note, we could move on to our second story here, and this one, Michael will bring us up to speed on totally drug-resistant tuberculosis in India.
Yeah, this is a truly frightening story. Totally drug-resistant Mycobacterium tuberculosis is not new. It was first described in 2002 in two individuals from Italy, and then it mercifully disappeared, and then it arose again. We saw it again in the literature in 2009 in Iran. And now, unfortunately, what we have witnessed is Mycobacterium tuberculosis, the totally drug-resistant variant, emerging in India. And the reason I bring this to everyone's attention is that Mycobacterium, the prevalence of Mycobacterium is the highest in India. For all countries in which tuberculosis is monitored, the nation that has the highest incidence of TB is, of course, in India. And for those of you who don't know about this disease, I'll give you a little bit of background. It's thought that Mycobacterium tuberculosis is definitely a pathogen of man. It's thought to have been domesticated at the same time, that is, began to live with man as an infection. At the same time, we began to domesticate livestock. And so when we began to domesticate livestock about 12,000 years ago, this acid-fast bacillus that is extremely resistant to drying became prevalent, and was transmitted. And the good news, bad news is you don't have to. It's not a highly infectious microorganism. That is, you're not going to stumble into it, but it's still a pretty good transmitter. It has an infection rate of about 22%. And for every one individual that is infected with Mycobacterium tuberculosis during the active infection, they're likely to transfer that infection to about 10 to 15 individuals per year. So you can see that it does, as they say, get around. And in a nation such as India, where you have a large cohort of physicians, and it's principally private physicians who have not been trained in the proper treatment of tuberculosis, where you have to use the right combination of drugs for the right prescribed period of time. You just don't go to a doctor and he gives you one pill and you're cured, because the hallmark phenotype of this organism is it grows so slowly, even in people. Garden variety E. coli, we all learned, had a generation time of about 20 minutes. Mycobacterium tuberculosis has a much longer generation time. Rather than measuring it in minutes, we often measure it in hours. With the under the best conditions of growth, it has about a 16-hour generation time. And when you begin to limit its nutrients and begin to throw the wonders of the immune system at it, it of course grows very slowly. And so oftentimes it's very challenging to go after this bug. The way it's transmitted, or the principal risk factor that individuals have of acquiring tuberculosis, is, of course, breathing, because the microbe is transmitted through the air on a cough, a sneeze, or your saliva, and it's typically done via droplet secretion. And you actually develop a productive cough when you have the disease, and the particles that your lung generates are often 0.5 to 5 microns in size. And we know from stories about 10 years ago when we were discussing anthrax, 0.5 to 5 micron size particles are energetic. They can remain suspended in the air column. So again, your risk factor is encountering enough of these particles, and then of course, because of their small size, they sink into your lung, and they typically infect the lower portion of your upper lobe of your lung, or the upper portion of your lower lobe of your lung. And the only good news in the story about tuberculosis is most infections result in an asymptomatic or latent infection, and then about 1 in 10 of those latent infections will sometime progress to active disease. And it's really remarkable the way the human body deals with this. This is one of these infamously cryptic terms like granulomatous focal infection. And what that simply means is your body begins to set up a wall. And if you look at a chest x-ray, and there's plenty of TB chest x-rays out on the internet, you can see how your body begins to wall it off with this granulomatous lesion, in which they recruit macrophages and other proteins to create this gonfocus, and that then becomes radio opaque, that you can actually see on your garden variety chest x-ray. But it really walls itself off, and the tuberculosis effectively just sits there walled off. And then, again, as I said, about 10% of the infections will actually progress to active disease. And this often occurs when you become a change in your immune status, whether you become pregnant, whether you become immune compromised. And the sad story is HIV and TB are truly synergistic infections that result in extremely bad outcomes for individuals that acquire both of those infectious agents. And so it's really bad. Please interrupt.
You do want to say that in the places which are blessed with good hygiene, this is a disease which is really limited to certain groups, people who are malnourished, people with AIDS, and so forth. It is not a source of great concern to the public health authorities among healthy people. Isn't that right?
That's what I was just coming to. I was going to point out that a third of the planet's population is infected. And to those of our listeners in the developed world, you probably don't know 1 out of 3 people have been infected with tuberculosis. And as Elio points out, the third of the planet that's infected are those in developing countries in which they don't have the same level of hygiene, they don't have the same level of public health vigilance
and nutrition, especially,
and nutrition. And it is a disease of nutrition. In fact, before the advent of antimicrobials that came to being after Selman Waksman in the 50s came up with streptomycin, which was the first true wonder drug to treat tuberculosis. People were often quarantined or sent off to sanitaria, and the treatment was rest and tremendously high diets in protein and nourishment so that they could deal with one of the hallmark symptoms of the disease, which is wasting or loss of weight that English literature writes about. I think Dickens writes about tuberculosis in one of his novels. I think.
Well, Magic Mountain is all about.
Yes.
Thomas Mann's Magic Mountain is all about tuberculosis. So.
Michael, I have 3 questions for you. All right. So if you have total drug-resistant TB, does that mean you're dead? There's no treatment then, right?
Well, it's hard to say because I think they're still trying to treat them. I think they're still trying to treat them, and they're mixing cocktails and whatnot. I don't know whether or not they're dead. I haven't found out whether or not those individuals in Iran have died. They were talking about the cohorts. There's a paper on the Iranian outbreak from 2009 that was much more extensive than the note in the letter to the editor in Clinical Infectious Diseases. And it doesn't describe whether or not the patients did indeed die. So I don't know whether or not they're dead. Right. The good news is that tuberculosis is a slow disease.
Right. But it's not always fatal, as you said. So these didn't necessarily die, these individuals.
The overall fatality rate for tuberculosis in general, when you have an active case of TB, is about 50%.
Oh, that's quite
high.
Substantial.
Yeah.
All right.
But it's chronic. I mean, this is over many years, right?
It's a chronic disease that I guess if you look at it like all the other chronic disease like HIV, if you happen to be infected with HIV, for tuberculosis in general, if you're infected at the time you acquire the tubercular bacillus, 30% of those individuals will develop active disease. If you don't have HIV, then the development of active disease is between 5 and 10% of the population.
All right, next, how do you get resistance to so many antimicrobials? Is it some kind of a pump that pumps them all out? Do you know?
Well, the drugs are extremely varied. If you look in the CID note, the first line antimicrobials are isoniazid or INH, rifampicin. So INH is, I learned it as an instructive antibiotic, where you effectively, it's involved in 1-carbon metabolism, if I'm remembering right. And it effectively makes the cell is unable to make something that the INH is serving as a mimic for, as is pyrazinamide, which is another first line drug that these microbes are resistant to, as is ethambutol. And so these drugs effectively are involved in basal metabolism, if you will, associated with the microbe. Rifampicin is obviously involved in messenger RNA. The second line drugs are things like moxifloxacin, para-aminosalicylic acid, azamine, kanamycin, linezolid, rifabutin. And then if you look at the genotypes of where the mutants were in the totally drug-resistant microbes, one was in the RNA gene RPO beta, which is in it was RPO beta mutant 3, the CAT G gene, which is the catalase peroxidase gene, gyrase A, which obviously is associated with the gyrase A subunit, which is in the rewinding of DNA. So the way you make RNA is you have to unwind the DNA. And then RSS mutant 1, and the RSS mutant 1 is, of course, involved in the 16S ribosomal RNA, and ribosomal RNA.
Multiple targets, basically.
Yeah, so there are many ways to resistance, and they all happen in these unfortunate people.
Yeah,
right?
I guess there's sequential acquisition of mutations, right? And
yeah,
you treat them inappropriately, and you select for these. Yeah.
Yeah, and that's effectively the argument. Our colleagues at NPR did a story that appeared on Morning Edition this morning, in which they interviewed Richard Chasen, and he said,
Richard Chasen being?
Richard Chasen is a TB researcher at Johns Hopkins, and the quote from Dr. Chasen is, "Total resistance is a new phenomenon that was entirely predictable." And he goes on to say in his report to Richard Knox, I believe it was the reporter who did the story, is that he tells him that total resistance is a new phenomenon that was entirely predictable. He argues that it's predictable because hundreds of thousands of people in India have what's already called multi-drug resistant TB, and a growing number have the extensively resistant TB, which is XDR TB. And of course, XDR TB was made famous by the individual who got married, got on a plane, went to Italy, and was positive for TB, and then he tried to sneak his way back into the country, and he was put into an direct observed therapy program out in Denver to try to cure his XDR TB. And that happened a few years ago. And so the issue is that what's happening in Mumbai, and this is where the total drug-resistant TB has emerged in India, and there's about a dozen patients out there. And what's so sobering, if you think about it, they all have active TB, and if you don't have drugs to control this microbe, you will continue to spread it. And if the spread rate is still 10 to 15 per year, you have 12 people infecting 10 to 12, and then each of those 10 to 12 people infect another 10 to 12 people. And the population density in Mumbai is pretty high, and the individuals often seek out private medical care in India, and they don't have they're not prescribing the right combination of drugs, or they're not checking to see what folks have, nor are the individuals taking the drugs for the prescribed length of time. Oftentimes, the therapy for XDR is well over a year, and I think they look at the individuals to see when they go smear negative in terms of being able to produce acid-fast bacilli from their sputum. So it's really quite sobering, and I think one of the recommendations of the WHO and many of the public health agencies that are worried about tuberculosis in our developing countries is where you can actually do it. They actually advocate direct observed therapy, where you watch the individual take the pill, you prescribe that they come back, and the direct observation of their patients really can snuff out epidemics. And we learned that as recently in the 1990s here in New York City, where we had the emergence of HIV and the drug abusing population, and then they actually became a focal point of active tuberculosis infections. And for many years, TB was, falling like a stone. And then all of a sudden, with the advent of HIV, we saw a resurgence in tuberculosis.
So is it likely that in X years, this strain will be global and quite prevalent?
I'm afraid that is time will only tell. Fortunately, we have HIV under control in the sense that it's now managed as a chronic disease. But still, if you have HIV, I'm sure the studies have been done to determine if you have zero viral load, even though you're HIV positive. I don't know if you're more susceptible to acquiring TB. I'm sure we'll get a letter to the effect telling us.
Yeah, it's because those individuals have higher CD4 counts, so they should have better resistance. But
knows? You never know.
You never know.
And so I'm really disturbed that I was always worried once we had MDR, and then when it became XDR, I was even more concerned. And now that we have TDR, totally drug-resistant TB, it's even more problematic. And I look at it from the fact that it's really how fast this microbe grows. The fact that it doesn't grow quickly really limits the activity of many of the antimicrobials, because after all, that's why they work so well in infections, is because they often are growing faster than our normal flora. In the case of TB, it's really just sitting there, idling in place when you have that latent infection. Or even in the case when you have an active infection, it's idling in place. It's not growing exponentially like Neisseria meningitidis does, where it's a fulminant infection. And so it's really hard. I think we need to be clever. And as I was thinking about the magnetosome paper and sequestering iron, I know that tuberculosis goes out scrounging for iron. So if we could starve it out by sequestering the iron, that may be a neat way of thinking about how we might be able to control this microbe.
May not be so easy to do because, you say, it grows slowly. There's not much of it. You'd have to deplete the whole body of iron before you can really be effective on TB. I don't know that that's so likely.
The whole issue of iron as therapy, and that's the fundamental problems. The bacteria are just so good. And unfortunately, since we carry oxygen around in an iron box called hemoglobin, we need a lot of iron because we have lots of iron boxes in our body called red blood cells to carry that toxic oxygen around.
All right. Thank you, Michael. Let us read a few emails. We have some interesting ones today. One is from Peter, who writes, Dear TWiM team, I saw this paper on how the variety of bacteria populating human skin can influence who mosquitoes chose for their blood meal.
Yeah.
It's paper in PLOS ONE, composition of human skin microbiota affects attractiveness to malaria mosquitoes. It appears that mosquitoes tend to avoid people with more diverse microbial communities on their skin, as they contain bacteria that produce volatiles that repel mosquitoes. It's interesting. So the less diverse communities, then there are more of them, and those individuals are more attractive to mosquitoes. So Peter writes, presumably once these particular bacteria have been identified, then it may be possible to culture them to produce a mosquito repellent. Presumably actually growing the culture on your skin would also work as biological control, although I think it would be rather harder to market given most people's fear of bacteria.
I think the interesting thing here is that it's probably those god-awful short-chain fatty acids that are stinky that the mosquitoes don't like. So I doubt whether or not people are going to want to apply short-chain fatty acids unless they're odorless to us.
Yeah, right. All right, thank you for that, Peter. The next one is from Jim, who writes, I saw this engrossing documentary about Lyme disease with a controversy about the existence of a chronic form. And he sends a link to that. The movie's several years old. Do TWiM panel members have any information about the controversy, and is progress towards resolution of what is affecting folks with chronic conditions they attribute to Lyme being made? Unfortunately, as noted at the end of the movie, several key researchers have died, including one who had detected Lyme borreliosis in brain tissue samples from deceased Alzheimer patients. I would have liked to have them join the TWiM panel and discuss current activities. Well, unfortunately, that's not possible. Do any of you know anything about so-called chronic Lyme?
Other than it's in the brain and there's an actual infection in the brain, that's the extent of the knowledge I have. I'm sure there's an emerging literature.
It's rather a controversial topic, right?
Yeah, I know it is, and I haven't followed it, so I couldn't add very much to it.
It was for what I could tell that, so I found two articles. One said the assumption that chronic subjective symptoms are caused by persistent infection with Borrelia is not supported by lab studies or controlled treatment trials. Chronic disease is a misnomer, and the use of prolonged, dangerous, and expensive antibiotic treatments is not warranted. And another review said some patients have chronic, long-lasting complications despite antibiotic therapy, probably due to adverse effects of the immune responses. There's some thought there are chronic symptoms, but perhaps not caused by the presence of the bacteria. Well, we should get someone who works on this to come on TWiM and talk about it.
Well, in general, the trypanomatosis and spirolysis are really weird kind of infections. I mean, they stand aside. Syphilis is the example of a disease that is more full of mysteries than anything I know. Mm-hmm. And it'd be nice to get somebody here who knows what they're talking about.
Yes, we should. Do you know anyone who works on this, Elio?
Not offhand, but we can think about it.
We'll find someone.
It would be a great Valentine's Day story.
That's a good idea.
Since syphilis is a disease of the desiratory tract.
Good idea.
Oh, yeah.
We'll target for February 14th. And our last one is from Varun, who writes, "Hello, everyone. First of all, let me congratulate the microbiology crew for bringing up such a fantastic podcast that makes microbiology so much fun. Since Dr. Moselio is very interested in the mitochondrion, I thought he probably would know the answer for my question. It's known that only mom's DNA is being passed to the progeny. It's also known that as you age, there is continuous accumulating damage to the mitochondrion thanks to the oxygen species. Now, my point is, why doesn't the mitochondrion, which is mom-derived, after a few rounds of replication, accumulate damage and pass to the child? That doesn't happen, else we already would have high damage in the mitochondrion. I hope my question isn't absurd." So what do you think, Dr. Moselio?
Dr. Moselio doesn't know. Sounds reasonable. I must say, I can't say that I thought about it, and I can't say that I know anything about it. So excuse me, I'll look into it. Okay. That's all I can say right now.
If you figure it out, I think you have the fountain of youth.
That's right. Well, let me look into it.
He also writes about an episode of HIV on TWiV. And then on episode 22, there was a great snapshot of microbiology in the clinic. I'm interested to know if Dr. Alfred has experiences regarding Burkholderia cases in infants. We just happened to see some in premature infants. If yes, please lighten the area. Sorry for too many questions and comments. Thanks again for the great EduCast. Never too many questions. So I wrote this to Al Sacchetti, our guest on TWiM 22, and he wrote, "To be honest, I've never heard of Burkholderia. So if I've seen a case of it, I never made the diagnosis." Al is our ER physician from number 22.
He's probably heard of Pseudomonas, and when he learned about Burkholderia, it was called Pseudomonas.
Okay.
And I think, I'm certain Dr. Sacchetti has heard of Pseudomonas.
Yes, I'm sure. He writes, "I looked it up, and it's more of an infection of the immune compromised, especially those with cystic fibrosis." There you go. Sounds like Pseudomonas, right?
No, it's different.
It's a different flavor.
Different flavor, okay. And a case in an infant would be particularly unusual from what I can tell. Seems more of a plant pathogen that also infects livestock and is more of an opportunistic human pathogen.
Certainly is an opportunistic human pathogen, but Burkholderia can be very heavy stuff. And I don't know much about it, but I really think that it's one of the emerging pathogens.
Really? It's interesting.
It is one of the emerging pathogens. it causes it's one of those microbes, Burkholderia mallei and Burkholderia pseudomallei. Mallei causes glanders, and it can be pretty devastating. And I think what we're going to learn in the years to come, as we become cleaner, we're going to see Burkholderia, because it's principally a soil organism, but once it gets into the rich environment of humans, it can wreak havoc.
Yeah, well, Al is a ER physician at quite a large hospital over in Camden. So was it surprising that he hasn't seen it?
I am surprised.
All right. On another note, I ran into a pediatric ER doc from Chicago last week who told me they are seeing one of the worst respiratory syncytial virus seasons ever.
Is that so?
Yeah, this has been our experience with more cases in the last few months than we have seen in the last 3 years combined. This is in the face of no influenza infections at all this year.
Yeah, we haven't seen very many influenza infections here either.
Interesting. Might be fun to do a benchtop to bedside show with an RSV virologist and a physician on this one. Happy New Year. There you go. Thank you, Al, for answering that. And that will do it for TWiM 25. You can find us, of course, at iTunes and at microbeworld.org/TWiM. You can get an app for your iPhone or Android device over there to stream the episodes. And please send us your comments and questions to twim@twiv.tv, or you can go over to microbeworld.org/TWiM and leave a question over there as well. Elio Schaechter is at Small Things Considered, a wonderful blog that you can find online. Thank you for joining us today, Elio.
My pleasure. My pleasure.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, Vincent. I'm going to go out to the pond, get some sediment, and look for those Magnetotactic guys tonight.
Hey.
You bring your magnetic stir bar with you, right?
Always. Never leave home without one.
So you have water that's not frozen down there?
Oh yes. It's not gotten cold yet. We had a little cold snap, and it's been pleasant.
If you do that, Michael, take your cell phone and take a little movie of it, okay?
Okay.
That would be fun. I'm Vincent Racaniello, and you can find me at virology.ws. Thanks to the American Society for Microbiology for supporting TWiM, Communications Director Barbara Hyde, Chris Kandian, and Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.